Thanks everybody for coming and everybody online. So today we have our Pioneers of Biomedical Science research seminar. And just before we start, I'll give a plug for next week. We will have Daniela Schiller from the Icahn School of Medicine at Mount Sinai, and she'll be talking about navigating abstract spaces. And we got a preview of that from Mike Phelan last night. But now it's a great pleasure to introduce a friend and colleague, Dr. Michael Bressan. People often say friend and colleague when they're introducing people. I actually mean it. Mike and I postdoc together at UCSF, and he's a extremely talented scientist. I'm sure you're going to enjoy today's talk. He's authored papers in New England Journal of Medicine, Circ Research, Developmental Biology, Science. I still remember the day he had that paper accepted. Cell Reports, Development Cell. He got his Ph.D. from Cornell in 2009. And before that, had been working on congenital heart disease and identified novel roles for myosins and some transcription factors in congenital heart disease. And he went on from there in his to get his PhD in Cornell in 2009 and did graduate work on the plasticity of cardiomyocytes and endothelial cell progenitor cells. Importantly, identified bone marrow cells having the capacity to differentiate into myocytes and how age-related paracrine signaling actually influences this process. And then did some really elegant work on vascular patterning and BMP signaling and generation of endothelial cells from mesodermal precursors. And it's then that at his postdoc in the McAllister lab at UCSF that I started to interact with Mike. And it's where he was working on developmental morphogenetic processes and how they set up the electrical system of the heart, right? So you guys know about the cardiac conduction system and the importance of that and the sinoatrial node. And so Mike's really contributed to our understanding of how all of this biology gets set up. And in doing so, he identified a previously unknown origin of the pacemaker cells and through systematic studies and revealed not just the source of these cells, but then did some really nice temporal and spatial work to understand how they differentiate, integrate, how the pacemaker cells differentiate and integrate into the heart itself. He's had his own lab at UNC since 2016. And here he has done some really nice detail work delving into the SA node further and identifying what cells are there, how they communicate, what extracellular components enable this essential pacemaker to do what it does. The work has implications beyond understanding really how the heart and its conduction system forms, but it's kind of contributing to understanding how electrical coupling itself works and really changing our understanding of that and how this may go awry in disease, therefore obviously how we may intervene if we understand this better. No surprise, he was awarded a K99 in 2014 and has gone on to obtain his own currently active R01 in his lab. He, like many of us, is not just taking stuff, but is serving and does serve on study sections to give back his time in that sense. And I have to say, he's one of the most dedicated and creative scientists I have the pleasure of calling a friend and colleague. I'm sure you'll enjoy what he's going to show you today. You're getting he's get ready for some really beautiful imaging. I think some of what he's going to talk about overlaps with the brains of some of the people in this room here. So should make for some good discussion. All right, hand it over to Mike.
We'll take my mask off as well. Microphone is working okay?
Yep.
All right, so I want to thank Jamie for that very thoughtful and nice introduction, as well as for the opportunity to come and share some of our work with everyone today. So as Jamie mentioned, I really view our lab as a developmental biology lab. We're interested in a number of aspects of different components of cardiac morphogenesis with really particular emphasis on electrical patterning in the embryonic heart. And so today, I didn't think I needed to do, let's see if this works. All right, I didn't think I had to give a whole lot of background information with this audience, but I just thought I would start with reminding everyone of a feature of the heart that I've always found particularly fascinating, and that's basically what we're looking at right here. So this is an embryonic chicken heart. It's got 4 chambers, the same as mammalian hearts. And basically what you can see is that even though this heart is completely isolated from the rest of the embryo, it beats, and it beats in a very rhythmic and coordinated fashion. And this is basically due to the fact that there is a system of cells within the heart that we broadly define as the conduction system, which is responsible for initiating the electrical impulse that drives contraction, as well as coordinating its propagation through the muscle so that the muscle basically contracts in the right order. And what we're going to talk about today, or the projects I wanted to talk about today, are really focused on this region of the heart right here, the sinoatrial node. which basically sits at the head of the whole conduction system. So this is where the impulse that triggers the entire cardiac cycle starts, right? And so the sinoatrial node is interesting for a number of different reasons. I think not the least of which is it houses a very unique type of muscle cell, right? So this is a cell that we call cardiac pacemaking cells. This is an actual cardiac pacemaking cell here that we've labeled a couple of different ways and we've recorded up. And basically you can see what this cell is kind of famous for. It's an oscillator. So here we're looking at calcium transient activity, but what you basically see is that it is pulsing on and off, right? It depolarizes and then repolarizes its membrane autonomously and rhythmically. And this is basically what gives the rhythmicity to the whole heart. But one of the things I want to kind of emphasize is that heart rhythmicity is not the product of a single cell, right? We don't have one cell in the center of the sinoatrial node that's driving the whole process. It's really an interesting feature of pacemaking cells, and that collections of these cells, and here we're looking at probably about 10 to 20 pacemaking cells, that have the ability to sort of synchronize their behavior, right? So all of these cells are collectively pulsing on and off, and they reside in this sinoatrial node. And so here we're looking at another imaging technique that we use a lot in my lab, voltage imaging. So what you're going to see is the signal here, which is the wave of depolarization as it propagates across the tissue. And really what is driving it is collections of these pacemaking cells that are synchronized with each other, that are initiating the action potential right there, that will then go on, propagate to the rest of the myocardium and trigger contraction, right? So it's really this rhythmic activity, you know, as we increase in scale, that is derived from the function of cells within the sinoatrial node, right? And so one of I think pacemaking cells and sinoatrial node are very interesting from a basic science point of view, understanding some of the concepts that are related to this type of behavior. But it's also a very clinically relevant area of the heart. So sinoatrial node dysfunction is actually very common in humans, co-segregates with a lot of different types of diseases, both cardiac and non-cardiac, as well as some other features that we think of as medically relevant. The reality, however, is despite how prevalent it is, we really have a poor understanding of the pathology. Right, so the actual cellular defects that underscore most forms of sinoatrial dysfunction are relatively unknown. It's kind of associated with fibrosis. There are a couple of channels and that mutations that are associated with, but in reality, what actually goes wrong in the vast majority of cases, we don't have a very good insight into. What is pretty clear, however, is that if you have significant dysfunction in the sinoatrial node, there is a clinical intervention that's required. That often takes the form of implantation of a mechanical pacemaking device. These are actually quite good and they get better all the time, but there are patient populations in which this is not an ideal approach, right? So you wouldn't want to go into a young patient and then have to repeatedly go in and change the position of leads based on growth or change batteries over time. So people have started to think about pacemaking cells in therapeutic sense, right? So can you actually construct cellular therapeutics, a biological pacemaker that you could put into a heart that could correct these types of chronic arrhythmias in patients where a mechanical pacemaker device is not ideal? But here again, you know, we actually lack a lot of understanding about sort of the cellular mechanics and the microenvironmental interactions that are actually required for pacemaking cell function. So we're missing some of the real critical design criteria that would be needed for that kind of approach to be really useful, at least for long periods of time, right? And so what we try to do in my lab is think about what's going on in development to try to inform us what the critical events are in patterning this area so that it is functional, where the vulnerabilities might lie, and then also try to understand what some of these criteria for future, you know, cellular therapeutics might be, right? And so kind of starting with this idea of, you know, trying to understand these cellular and microenvironmental interactions. For our purposes today, I kind of broke this talk into pieces, right? Where I just want to stress that we can think of pacemaking cells as a very unique type of myocyte that resides in a very unique microenvironment in the heart, right? And so I'm going to put up these are scanning electron micrographs through a human sinoatrial node and a human ventricle. I think even if I didn't explain anything to you, I think most people can appreciate that these are very different muscle beds. If we kind of break this up, you know, and think about it in terms of a niche, right? We have the sinoatrial node has very long been known to have high levels of interstitial extracellular matrix. There are lots of non-muscle cells that reside in this area of the heart. It's heavily innervated, and it's usually thought of as being fairly isolated from the working myocardium. When we think about the cells, and here I'm just isolating a single pacemaking cell versus a single ventricular myocyte, you can see they're very small. They tend not to have any axis of alignment like these beautiful working myocytes here, ventricular cells. They lack these large electromechanical junctions that you see throughout the rest of the heart. And they have other features I've just highlighted here. They tend not to have very many sarcomeres. They can contract, but nowhere near as efficiently as ventricular myocytes, right? And so today, what I was actually going to do was kind of present some more evolved or completed work that we've done, looking specifically at what's going on with the cells, how they kind of figure out how big they need to be, how they start to figure out how many ion channels they need to express, things like that. But kind of at the last minute, I kind of changed my ideas here. And what we're going to talk mostly about today is this niche, this idea of the microenvironment that's built around pacemaking cells and how that actually influences their activity. Right. And so, yes, in general, what we really are looking at is how are all these features established and how they influence activity. And we're going to focus on the niche. So, thinking about the microenvironment here is something that's been of interest to me for a long time, going back to very early stages of my postdoc, but we really didn't have the tools or the insight at the time to really look at it. So when I started my lab a few years ago, we really thought, let's approach this from very first principles analysis. So, asking some very simple questions. So when during development does this niche form? What kind of processes control sinoatrial node morphogenesis? And then coming back, we always come back to this idea of what's the functional consequence of all these things that are happening. So in the next couple of slides, I'm going to try to take these in order, starting with the idea of when during development you start to see the sinoatrial niche form. So what I've outlined here is stages of cardiac development. This is again in the chick. And what we're going to focus on is this stage here. So during my postdoc, one of the things that we figured out is this is about the first stage in development in which cells that will go on to become pacemaking cells actually become electrically active and start pacing the heart, right? They're located just outside of the atria here in this little structure there. And so one of the first things that we did was basically go through and cut thick sections through this junction of the forming sinoatrial node and atria, and basically look at the architecture of the tissue. So this is what one of these sections looks like. I can rotate this around for you. And basically what you can see, the muscle layer here is labeled with MF20 in green, is that this is the forming sinoatrial node or pacemaking region of the heart. It is molecularly distinct from the atria at these stages. It is expressing different things, but structurally it looks almost identical to all the other muscle in the heart. There's nothing there that triggers any sort of impulse that this is a different area. We actually surveyed across development starting from here. And what I'm going to do is skip a couple of days. This is actually 3 days later in development in the chick. So now the heart is structurally quite different. We have the pacemaking cells basically located here, and that will eventually trigger an action potential in the right atria. We basically are taking the same approach. We cut these thick sections. And what you can see as you rotate around this now is that the forming sinoatrial node, which is located here, looks very, very different from the atrial myocardium. So you can see that the wall of the heart is much thicker here. And even at this fairly low resolution, you can see that the muscle here is very mesh-like, right? It's nowhere near as condensed as what you see in the atrial myocardium. We can look at this in a slightly different way. Here, we're just using standard histology. And what you can see at these very early stages, pacemaking cells and atrial cells look very similar. Whereas just 3 days later, now pacemaking cells are kind of broken up into these discrete clusters with things like collagen being wrapped around the clusters. And this morphology looks very different from the atrial myocytes at the same stage. Right. And so what we became very interested in at this point is we've kind of defined this little developmental window where something is happening which really fundamentally changes the structure of the sinoatrial node. So we're trying to figure out what that process might look like. And so this is actually work that was published a few years ago. So I'm going to summarize it very quickly. And what we basically came to was the idea that this remodeling process was actually not due to the pacemaking cells themselves, but was actually the product of pacemaking cells integrating with cells that came from this structure here called the proepicardium, which I've labeled with TBX18, and here in the diagram, right? And so traditionally, what we think of as the proepicardium doing is it's a population of cells that sort of binds to the myocardium and spreads across it, giving rise to the outer layer of the heart, the epicardial layer of the heart. But what we found is that during this process, or actually even preceding this process, large numbers of cells leave the proepicardium and actually invade the underlying pacemaking region of the heart and remodel that area of the heart, which was, a fairly crude set of experiments, but gave us insight into a lot of different processes that might be taking place here, and also gave us the ability to do some very classic experimental embryology, and that is basically remove the proepicardium before this remodeling takes place and ask what actually happens to the structure of the sinoatrial node and how it influences the function of the sinoatrial node. So in the chick embryo, this is basically a process of going in and just pulling it out and then allowing the embryo to continue to develop and looking at the sinoatrial node at progressively older stages, and in this case, up until it caused lethality. And so I'm going to go through some of this data. So I showed this a few slides ago. So this is basically whole mount voltage imaging of the heart, right, with the sinoatrial node and the right atria. It turned out for this analysis, it was actually easier to quantify and get an image of what was happening by cutting thick sections and doing something closer to what a brain slice would be, right? And so this is a slice through this sinoatrial node atrial junction where you can see an impulse start in the pacemaking cells, propagate through that hinge, and then move through the atrial tissue. So this is in fact a sham operated embryo, right? So, or a control embryo for our proepicardial ablation studies. And basically what you can see is this looks very normal for what we would expect, right? So we have an impulse that starts in pacemaking cells, it moves around and excites the atria. These are the wave traces of the action potential. You have very pacemaker-like action potential. They're very rhythmic. They precede atrial action potentials. And this is really kind of focusing on the upstroke of the action potential. So you can see an impulse starts in the pacemaking cells and then kind of fights its way around that hinge into the atrial myocardium. Now, when we got rid of the proepicardium, right, before this remodeling process took place, we actually saw a number of fairly complex types of electrical phenotypes, but probably the most prominent was a form of conduction block, right? So an impulse would start in pacemaking cells, but it couldn't get through this hinge and couldn't excite the atrial tissue. The pacemaking region also became less rhythmic. And here you can see, again, looking at the upstroke here, that the amplitude of the action potential is dropping really dramatically as it's trying to get out of the sinoatrial node. Now, we didn't know much beyond that at this point, but we decided to actually look what was happening structurally. And so this was actually pretty interesting. So again, this is a sham. It looks pretty much what I showed you a few slides ago. This is what happens when we do the ablation. And you can see that the myocytes, the pacemaking myocardium, has condensed now. It's very compact and very different in structure than what we see in the normal sinoatrial node. This is just another way of looking at that. Again, this is a 3D reconstruction through the sinoatrial node where we have these mesh-like cells that are very loosely interacting with each other, and they condense down into these fibers like we would see in the atrial myocytes when the proepicardium is removed. And then there are some molecular changes that we also see. This is the gap junction connexin 40, which we usually do not see at high levels in the sinoatrial node. And when we got rid of the proepicardium, we started these strands of cells that were activating connexin 40. So there's a bunch of structural and molecular changes that are taking place, right? So that basically all that did is tell us that whatever this process is, it is important for sinoatrial node function and the ability of this tissue to take on its mature properties. And that's kind of where we ended this study. So I'll just kind of summarize it for you here. So this is a 3D reconstruction of the sinoatrial node. These green cells are from the proepicardium. The red cells are pacemaking cells. And so what we kind of concluded was this remodeling process is actually active. It requires cells invading and interdigitating with pacemaking cells. The cells come from the proepicardium. And if you block that process, the whole system kind of falls apart. The question that we were left with at the end are basically, well, who are these cells and what are they actually doing? Why does the sinoatrial node need them, for lack of a better term? And so now I'm going to transition into some studies that are ongoing in the lab, and some of them are fairly new.
That's a question before you transition.
Sure.
Does the proepicardial tissue need to remain, or is it a very narrow window within that time frame you study that the appropriate signaling is done, or does it have to stay there beyond that?
In our hands, if you don't get it at the right stage, the tissue recovers just fine, right? So the way that we think about that is you have to remove it before the integration process starts, because otherwise, if you even start to get some cells in there, they proliferate enough to recover, right? So I don't know what would happen if you went back and later tried to ablate them, but that is where we're kind of thinking about this in terms of disease modeling, that these cells, if you change their phenotype, that you might be influencing what happens in the adult and causing disease. I hope that answers. Yeah, okay. So what I'm going to talk about for almost the rest of the talk is really work that's being done by a postdoc in my lab, Marietta Easterling. And she started right before the pandemic started. And she's actually made a decent amount of progress considering how difficult times have been over the last year or year and a half. And so she basically came into the lab with this being the underlying question of what cell types, what are these non-muscle cells and what are they doing? And so one of the first things that Marietta did is she basically went into the heart at these stages towards the end of what the remodeling process that we saw was taking place. She isolated the sinoatrial node, the free wall of the atria, the right ventricle, and in fact, she took some AV tissue as well. And she prepped it for a 10x genomics sequencing study. So basically a single cell sequencing study. Now, we're pretty new at this. So, we're still kind of working through the best way to analyze this data. But some very interesting things popped out almost immediately that I kind of want to show. This is basically her concatenating all of the cells that she took from all the different regions and actually clustering them by cell types bioinformatically. And I'm just showing this to show that there are lots of different cell types in the heart at this stage. But when you actually overlay that on where the cells came from, you can see that there are clusters here that are very specific to the sinoatrial node. And when we actually started looking at the cells specifically that came from the sinoatrial node, what we saw is there's, again, lots of different cell types. I think there's 11 or 13 different cell types that we've characterized so far. There's different types of muscle, there's different types of fibroblasts. The epicardial layer is actually quite unique in the sinoatrial node when you compare it back to the rest of the heart. And so what we're doing now is a lot of pathway analysis and trying to figure out the best way to sort of pull data out of this. But one of the first things that we saw was actually kind of surprising to us. And so here we're looking at one of the muscle clusters. We've arbitrarily called it muscle cluster 2, but we think this is the pacemaking cells. And these are the top 20 genes that were used to call it as an independent cluster based on the single cell sequencing. This is the same for one of the fibroblast clusters, right? And so a lot of these genes are not surprising. But what we ended up seeing is BMP2 is one of the highest expressed genes in this pacemaking cluster. And when you look around, what you find is a lot of these other clusters, this one was actually just a very nice example of it, are showing prominent expression of a lot of the BMP target genes that are in the literature, right? And that really caught our attention. We didn't really need to do any advanced analysis to see that. The other thing that we saw is that when you actually start to look at the genes that are really upregulated here, they overlapped really well with a gene regulatory network that's already fairly well described in another area of the heart. This is where the endocardial cushions will form, and I'll get into that in a second. So these things really caught our attention because we also know that BMP2 is a critical component of endocardial cushion patterning during development, right? And so what we've been doing is going through and validating this, trying to make sure that what we're pulling out of the single cell sequencing is actually expressed where we think it is. This is RNA scope, so this is mRNA for BMP2. You can see it's expressed in the sinoatrial node. It's counterstained with DAPI here, and it very nicely labels what we think are the muscle cells in the sinoatrial node. Now, as I mentioned, we had seen a bunch of genes that we associate with endocardial cushion formation. So for people who are not cardiac embryologists, the cushions are these kind of cool structures that form in the developing heart. They are populated with this material that we call cardiac jelly, which basically sits in the basement membrane between the endocardial layer and the myocardial layer. And these things serve as kind of primitive valves in the early heart to prevent blood from kind of moving backwards during contraction. And we know that BMP2 is actually very important for cushion formation, right? This is a paper from just a few years ago indicating BMP2 is important for extracellular matrix, hyaluron and versican deposition in these AV cushions. Now, the fact that we had all these AV cushion genes upregulated in our RNA study was interesting because if you actually look at the sinoatrial node, there's no cushion, right? This is the endocardial layer and this is the muscle layer, right? There's nothing like a cushion that forms in this area of the heart. Or if you zoom in, and here we're staining with Alcium blue, which labels hyaluron, among other things, what you actually see is that there is a cushion-like matrix that looks like it's forming in the spaces between myocytes, in these gaps where, it's not nearly as robustly expressed in the atria at these stages. So we've gone through and actually kind of cross-reference what we're seeing in our RNA sequencing with what's in the cushions and what's in other people's sequencing, and starting to characterize what we think is going on in these interstitial spaces between myocytes, or what actually we thought, and when we started was going to be the interstitial spaces. This is tenacin C, another extracellular matrix protein, and you can see it's actually very enriched in the cytometral node, and it's very tightly associated with the pacemaking myocytes. This is aggrecan, another sort of member of this type of matrix that we're seeing enriched in these spaces between pacemaking cells. And so as we've been doing this, we've kind of been taking a much closer look at not just what's being made, but where it's being made, right? So here we're looking, this is actually a super resolution image of the cytometral node versus the atria. We're staining with WFA, which labels a number of different chondroitin sulfate proteoglycans. And what kind of popped out at us is you have these clusters of pacemaking cells that are surrounded with this WFA positive material. And this looks very different than what we see in the atria, right? So in the atria, it's just associated with the endocardial and epicardial layers. And if you look even closer, you can see some other interesting things. So I'm going to point your attention to this guy here. So this is a cell. It is not MF20 positive. It's not a muscle cell. It is very highly positive for WFA, and it's sending these extensions which hit the surface of this cluster and spread over it. In fact, here's another one, which is kind of hugging this cluster and this cluster. Here's another one, which is sending extension, and there's another one down here where you have these little feet that are WFA positive that are sitting on the surface of the pacemaking cells. If you zoom in and look at this a different way, this is now a transmission EM. The center of our field of view is a pacemaking cell. And these guys, which are much more dense in ribosomes, and so they appear darker, are what we think are these fibroblast cells, which are basically surrounding and hugging the pacemaking cells. And in fact, even at this resolution, you can see all of this extracellular material that's being deposited on the surface of the pacemaking cell. And this looks very different than what we're seeing in the atria at the same stage. So these are a pair of atriomyocytes, and there's this little basement membrane-like extracellular material that's being deposited here. The endocardial layer of the heart would be out here. This is the pacemaking cells, and you can see all of this material that's being deposited on the surface, which I'll just highlight there. And in fact, that and there's one other that are actually sort of little fingers that we're seeing projecting from what we think are the fibroblasts. So what we started to think about here is whether or not the interaction between the fibroblasts and the pacemaking cells is actually building a very unique paracellular type of matrix around the pacemaking cells. That's what we started to think about. And in fact, we went back to the single cell sequencing and started looking at who's expressing what, right? And so this is one of our muscle clusters here. You can see it's expressing BMP2, but it's not expressing any of these other genes at high levels. This is one of our fibroblast clusters, and it's activating transcription factors, MSX2, RUNX2, PRX1, which we associate with proteoglycan matrix production in the bone. And it's also making a number of these types of chondroitin sulfate proteoglycans. In this case, I'm showing you Bressan. So we started to think, okay, well, this is an interaction between these two populations, which is building a very unique matrix. So again, we went back to what the composition of the matrix that we were seeing built was, and we started looking at the literature, right? You see these kinds of things in the bone, and like I said, in the cushion. But the other place that you really see it is in the nervous system, right? And so the molecular composition that I'm showing here, this is from a review a couple years ago about these structures called perineural nets. And they wrap around the soma of fast spiking neurons in the brain, right? They have hyaluron backbones, they have chondroitin sulfate that are cross-linked into the hyaluron, and then you have tenacin, which basically stabilizes the whole system. And almost to a molecule, we're seeing the same things expressed in the forming cytometral node. Now, the reason that this caught us our attention is because this is not some just inert matrix that's there for structural integrity. In these perineural nets, these are thought to act as ion diffusion barriers. They can modulate membrane capacitance. They can limit the formation of synapses, and they also can control ion channel localization. So we thought, well, this is a really interesting matrix that's forming. It's forming right on the surface of the pacemaking cells. Let's see if we can manipulate it and see what happens. And so what Marietta started doing was very simple. So she basically started introducing enzymes into the system that would degrade this matrix, right? In this case, she did it with hyaluronidase and chondroitinase, two separate approaches. The data is basically the same for both. I'm going to focus on the hyaluronidase today because we have more numbers. Well, I'll get into it in a second. So for people who are not familiar with the chick as a model, I'll just kind of show what an experiment like this looks like. So we have the ability to basically window the egg, and we can develop to whatever stage we want. And then we can introduce or manipulate the embryo in a number of different ways. In this case, we're injecting the enzyme directly into the pericardial space around the heart. And then this was designed as a time series experiment where we pull tissue out at different stages and look at it. And today I'm going to talk about the 6-hour time point. So everything you're going to see is 6 hours of hyaluronidase treatment. And basically the approach is very similar to the single cell approach, where we isolate the synovial node, the atria or the ventricle, and then instead of dissociating and looking at what's being expressed, we actually prep these for imaging. In this case, it'll be voltage imaging. Right. And so this is what the voltage imaging looks like. On the top here, this is a vehicle-treated atria. This is a vehicle-treated synovial node. This is the electrical impulse basically initiating and moving through that little piece of tissue, right? And we can do lots of things here. And what we're showing here is basically the action potential wave trace from atrial and synovial node. So this is the control. This is basically the baseline that we would expect to see. Now, it turns out when we treat with hyaluronidase, and remember, we're treating the whole heart with hyaluronidase in these studies, we find that it has almost no effect on the atria. Everything that we look at looks normal. Action potential kinetics, conduction velocity, upstroke potential, everything looks fairly normal. So it has very little effect. However, the synovial node is actually really sensitive to this, right? And so you can see that this is very odd looking. This is one of the only ones where we can see in the voltage imaging an actual change in fluorescence. So that's why I picked it for presentation purposes. But you can look at the amplitude of the signal is very different than what we see. You can see the action potential shapes look different, and there's a lot more noise in this recording. And in fact, this is another example, not this heart, where we're looking at two traces in the same heart, we see all of these little tiny spikes that don't align with each other. And this is pretty common in all of our imaging. And so our initial thought was that these might be little electrical impulses that are propagating within the tissue that are just not synchronized with each other. That was our initial thinking. And so our initial conclusion there was that what we're doing is we're breaking apart this matrix, and the electrical communication between pacemaking cells is starting to fail. And so from here, I want to take a little aside, because I think most of the time when we think about activation of the heart, we think about conduction, right? An individual cell depolarizes, it creates an ion gradient with a neighboring cell, the ions move, and then the second cell depolarizes, right? So there's kind of this pearls on a string type way of thinking about the way an action potential moves through tissue. It's a little more complicated in the synovial node, and that comes down to the fact that pacemaking cells don't innately have the same rate of activity. So this is clusters of pacemaking cells that we've isolated enzymatically, so trypsin and collagenase. And what you can see is they're not pulsing at the same rate or at the same frequency. And this is pretty common, right? And so you can imagine that just kind of squishing these cells together, you're going to get a lot of complex electrical activity. And it turns out that each one of these clusters is actually rhythmic in its behavior if you chart it, but that the rates are different. And in fact, the kinetics of the calcium transients are actually different, right? So the upstroke is fairly consistent, but the clearing of the calcium is very different, right? So it would be very, very hard to get a cell that has these characteristics to synchronize with a cell that has these characteristics, just because they would be completely out of phase with each other. And so sort of the mathematics of taking oscillators like this and forcing them into synchronization is actually pretty interesting. But to kind of summarize it, the way we think about it is that it's the amount of information shared between the independent oscillators that actually will bring them into phase, right? And so I just wanted to kind of give that as a backdrop of synchronization being slightly different in the way we think about it than conduction. All right, so our voltage imaging was actually too low resolution to actually be able to feel confident that this was a synchronization issue. So what we ended up doing was turning to another system that we use a lot in my lab, and that is to basically create mosaic hearts. We use a combination of integrating DNA plasmids and lipofectamine-based chemistry, and we can introduce things to genetically modify populations of cells within the tissue. In this case, we're introducing a membrane reporter and a calcium reporter, right? And so this is what this looks like, where you have a heart where the membrane's labeled. You can see the calcium transient in those labeled cells, and this is just the overlay. And we can use this system to basically ask what individual cells are doing in the tissue at any given time. And so, again, we can go through and isolate the synovial node and basically look at the calcium transient in different populations throughout the tissue and in sort of a mosaic fashion. So it turns out when we do a vehicle treatment, everything looks fine. So this is atrial trace. Each one of these colors is an individual cell. So this is, I think, 6 to 8 cells in the atria and an equivalent in the synovial node. And what you can see is these traces basically overlay, suggesting that all of those cells are oscillating in unison, right? And the amplitude is different, which has to do with kind of the focal plane that we're dealing with. But you can also see that the transients are about the same width, right? So they look like they are entrained in multiple ways. When we look at the hyaluronidase treated samples, we see that the atria looks again fine. But when we look down at the traces from the synovial node, we see lots of different independent oscillations that do not seem to coordinate and condense into one synchronized impulse. So we're starting to feel very confident that what we're seeing when we get rid of that matrix is a loss in synchronization. And this is kind of just to show you that treating with hyaluronidase looks like it's doing what we think it's doing, right? So this is vehicle-treated atria and synovial node. You can see there's a lot of material here. This is WFA staying again on the surface of the pacemaking cells. And we treat with hyaluronidase, we're kind of stripping that away. What is not happening is that the tissue is not falling apart, however. So we're staining here for a marker that we like to use for pacemaking cells. This is FLRT3. This is the synovial node. And you can see the cells are still in close proximity to each other. The tissue architecture looks fine. It's not like we're dissociating the tissue, but we're getting some sort of breakdown in this matrix, and it looks like it's causing the electrical defects that we're seeing. Okay, so I'm going to summarize that really quickly, right? Because that was a lot of information, I feel like. So we started off with trying to understand what's going on with this niche. And through our single cell analysis, and I didn't talk about it at all today, what we're seeing is that a fibrillar matrix, collagen, elastin, things like that contribute to the tensile properties of the tissue is being built. But sort of below that, there seems like there's another process taking place here, which we're thinking of as sort of a division of labor, where pacemaking cells are specifically activating a matrix program in the adjacent fibroblasts. And these fibroblasts are laying this matrix down around pacemaking cells. And if you get rid of it, the system starts to break down. And so we were actually really quite interested in this, and we continue to be. And so I'm going to go back to this idea of synchronization, because this is where our thinking really went for a long time. And the way that we think about synchronization in pacemaking cells is that the information that's being shared between the oscillators is being shared through gap junctions, right? That's the classic model of how this works. So you have two independently oscillating systems, you link them so that they can share information, in this case through a gap junction, and that's how they actually come in synchronization with each other, right? So you have these sub-threshold current flows that are going back and forth, and eventually the two cells come into phase with each other. And in fact, if you go through the literature, you can see that there are different states of synchronization that can occur based on the conductance of the gap junctional coupling, right? So at fairly low levels of conductance, you can get cells to come in phase with each other. And if you continue to increase that conductance, you will eventually come to a point where even if they have different calcium kinetics or voltage kinetics, those will match up and line up as well, right? So initially we thought, all right, well, we have to be messing around with the gap junction here. We thought something about that matrix stabilizes the gap junctions that are present in the synovial node. And we had all these experiments lined up. We were going to track how the gap junctions move when we break this matrix up. And we're still going to do those. But one of the things that we kind of backed up and said, all right, let's look really closely what the baseline looks like. Where are the gap junctions? Where are they expressed? And how would we be manipulating the gap junction content in synovial node if we started changing those things? And so I'm going to kind of get into that in the next couple of slides and really start to characterize the distribution of the gap junctions in the synovial node. Just as a reminder, this is when we're looking in tissue, so that you believe me that this is the synovial node. This is where the impulse starts in sections. Initially, one of the things that we did is just looked at transcript levels. We have a number of different sequencing data sets in the lab that we referenced. And just really quickly, what we saw is if you're looking specifically at the synovial node, the most abundant gap junctions are connexin 40 and connexin 45. Even though you have very low expression in the synovial node, those are the two most abundant transcripts. So when we started looking at these things, this is an antibody stain for connexin 40. Staining for these things in the chick heart is actually quite challenging. This is some of our better data. You can see, though, however, there are gap junctions in the atria, which we expected, but we actually have a very hard time detecting any connexin 40 positive material in the synovial node. Now, this is very likely or could be due to limits in resolution in terms of seeing this stuff. So we actually said, all right, well, let's go back and look at the mRNA. I think this is out of order. Anyway, so what we're looking at here is a cropped image of this intersection between the synovial node and the atria. This is a separate heart, but we've just imaged it at higher magnification. And that video played very quickly, but I'm going to explain what you actually are going to look at here. So there's this core region of the synovial node where you're going to see the impulse basically comes up all as one time, right? You don't see any propagation. The whole thing just comes up. Then you'll see rapid propagation, which is actually along the back of the inflow, away from the heart, and then slow propagation towards the atria. So I'll play that again, and hopefully you'll be able to see that, right? So that all came up. You had rapid propagation this way and slower propagation this way. So I'm showing that just to be able to superimpose onto it where the gap junctions are actually being expressed. So this is an image. Again, we're using RNA scope. This is for gap junction alpha 5, connexin 40, right? So you can see that it's expressed in the atria. It kind of ramps up at that hinge and then increases as you move into the free wall of the atria. It's very low in this core region of the synovial node where everything is coming up at once. And then it rapidly propagates here where we have some of that connexin 40 expressed along the back of the heart. Right? Now, we kind of expected this based on the literature that we'd have very low levels of gap junction transcript here. That's not new. We were kind of expecting that. What we were actually surprised by was how low they were. So this is RNA scope. So each one of these puncta is supposed to be an individual mRNA species, depending on how much you believe that. When we actually looked at this, and now this is connexin 40 and 45 and connexin 40. When we actually looked at this in detail, what we started to see is there are large populations of cells within this core component of the synovial node where we see no gap junction transcript at all. And in fact, we went through and we tried to quantify this a little bit more systematically. We arbitrarily picked these regions that we compared across different embryos. And this is the quantification. It's a little bit complicated. So this is a logarithmic scale, and this is RNA scope, so spots per nucleus. This is 1, so this would be 1 spot per nucleus, meaning each cell has 1 transcript level. And what you can see here is particularly in these regions, which are the central region where everything is sort of synchronized, what we see is many, many fields of view where we have less than 1 RNA scope, or in fact, less than 1 RNA scope per 4 or 5 nuclei, suggesting that there are a lot of cells in here that are not making gap junction transcript. And to us, that didn't make sense. How could they be in synchrony with each other if they're not even making the transcript? Yeah.
Well, kind of a stupid question, but where's the connexin 45 transcripts? And that's the larger map you've got up at the top of that thing.
Yeah, so it kind of mimics the 45, it's just at lower levels. Yeah. In the atria, there's some interesting differences in 40 and 45 that we see, particularly around the muscle bundles of the atrial wall. But in the synovial node, you have a little bit higher 45 here, but you can kind of see it in the quantification here. It kind of lags behind the 40 at lower levels, but it's a similar pattern. All right. Okay. So, kind of merging this with what the field believes is how synchronization occurs was tricky for us. And in fact, we thought about this for a while, and actually we're kind of inspired by some of Rob and Steve's work here, and started to ask questions related to, well, do these cells actually need gap junctions? There's lots of stuff in the literature about knocking gap junctions out of mouse models, and you can see that certain areas of the heart are not affected at all, and synovial node is one of them. And we thought, well, that would match up really well with this idea that you may not actually need gap junctions that much here. So we basically started asking this by introducing pharmalogical inhibitors, right? And this is basically a control experiment that we ran with one of these gap junction inhibitors, beta glycerotinic acid. Basically, we plated atriomyocytes on a micropattern cover slip, so they're all sort of aligned with each other. And then we perfuse in the blocker over time, right? So this is pre-treatment. You'll see an impulse that moves very fast across the culture. This is within 2 minutes of BGA, right? You can see that everything is desynchronized and you get these very slow action potentials that are propagating in the culture. So we thought, okay, great. This is a good reagent. Let's see what happens when we put this on our explants. And when we do that, what we see is pretty consistent with what we thought, right? So this is pre-treatment. This is an atrial explant, an action potential initiated here and propagated there. Here's 5 minutes after treatment, and this is an isochronal map. So sort of the color gradation is telling you how long that it took the action potential move. But we see a slowdown in the action potential propagation. Now, here's what we see in the sinoatrial node, right? So there's this central region of the sinoatrial node where the action potential initiates and propagates, right? That area basically is completely unaffected by BGA, right? However, on the periphery here, you can see it actually slowed things down, right? So this was telling us that, you know, whatever is going on in this core region of the sinoatrial node doesn't look like it's sensitive to BGA. Now, all of these uncouplers have secondary effects. So we basically went back and asked what happens if we tried another one, an unrelated one. And here we're using CVX, and we see basically the same data, right? So here's an action potential in the atria that slowed down pretty dramatically. When we look at the core of the sinoatrial node, the initiation height shifted slightly, but the actual spread here is very similar before and after treatment, whereas it slows down on the periphery. Sinoatrial node. So we're pretty confident that these reagents are actually working. But what we're seeing is that whatever is happening in the central, the core of the sinoatrial node, doesn't seem to require much coupling, right? And here's just some of the quantification that we've done, right? So here, this is the CVX treatment, and this is time of treatment. Conduction velocity, you can see there's a dose-dependent and time-dependent slowing down in the atria. And then we don't see any of that in the core of the sinoatrial node. And here we're basically just plotting sort of the endpoint for each one of the reagents that we've used. And you can see sinoatrial node conduction velocity doesn't change much, whereas it changes very dramatically in the atria, right? And so, you know, this is kind of where we are with this project, where we've been thinking about this. So we know that the sinoatrial node doesn't have a lot of these gap junctions expressed, and we know that it's very insensitive to these forms of blockade. And so from here, I'm going to wander a little bit into the realm of speculation here and give you an idea of what our current working model is. And so here what we have is two pacemaking cells. This is the cleft between the two pacemaking cells. And here you can see one of these extensions from one of these fibroblasts, which is basically sitting right in the mouth of the cleft. We can zoom in on that, and you can see all of the material that's being deposited on the surface of the cluster. But as we kind of look at the cleft, you can see that none of that material is being deposited in the cleft. And in fact, the spacing of this cleft is really tight. We see it's often under 30 nanometers. And in fact, in this, you know, this is probably closer to 20 nanometers in a number of different spaces here. So the cells are in really close proximity, and they are wrapped by a charged matrix, right? If we look at this from the staining point of view, and again, this is the super resolution imaging we did with some of the markers. Here we have a couple of pacemaking cells that are in close proximity. Surrounding them are these fibroblasts that are very positive for this WFA positive material, these extracellular matrix proteins, which seem to wrap and it's almost eat the pacemaking cells, to be honest with you. And then if you look here as well, I haven't had a chance to introduce this. This is another molecule that we've been studying a lot in the lab, FLRT3, which is an atypical adhesion molecule that you see in synapses in the brain. And it really likes to hang out in these clefts between cells, right? And so the model, or the way that we're starting to think about this, is that pacemaking cells exist in this configuration where you have membranes that are very closely opposed to each other. They use these kind of neuronal atypical adhesion molecules to keep those membranes in very close proximity. And then you wrap the whole system in a paracellular matrix, right? And what that does is it limits free ion diffusion with the interstitial space. So as an individual cell depolarizes here, it changes the ionic concentration in the cleft sufficiently where it influences the memory potential on the adjacent cells. So some of the olfactic or field potential concepts that I think Rob and Steve have been working on. And even though gap junctions may form or may not form in here, this is robust enough that if you lose a gap junction transiently or you inhibit it, like we've been doing in our studies, it's enough information shared between the two cells that they keep them in phase. And that if you destroy the matrix on either side, it disrupts the system enough where cells can start to fall out of phase. So really this highlights this matrix particularly as kind of an interesting component of the cytonutritional node that has not really been studied and may influence the activity of the tissue very dramatically. It also kind of gives us some insight into what types of communication might be taking place between pacemaking cells. So like I said, that's a working model. So you'll please forgive me for some of the speculation in there, but I think that's where some of the studies are going. I'm probably running late and I apologize for that, but this is who did the work. So most of the work here was done with Marietta. She's had help from a clinical fellow, Tony, and two technicians, Trevor and Julie. These are some of our collaborators and some of our resources and some of our funding. So with that, I'm happy to take any questions. Yeah.
You can answer that. Please. Steve, would you like to ask a question? Yes. That was absolutely beautiful. And something that's always bugged me about pacemaker cells in isolation when we teach this is the HCN channel. It simultaneously conducts sodium inward and potassium outward. And based on the slide, the last slide was your speculation slide, I too have speculated that maybe actually HCNs are localized at different portions of the cell. Do you have any evidence that HCNs that there's a population between in these classes and outside these classes?
Yeah, so that's an excellent question. So, and it's actually one that we're really interested in. And that project in our lab is actually a little more mature than this. So what we have found thus far is HCN likes to hang out with caveolin, right? And when you actually change the mechanics, right, you start pulling on the cells and you're stretching the cells, caveolin start to flatten out and HCN starts to wander. And so the way that we're actually thinking about this is these invaginations or the caveola that sit in the around the cleft. And in fact, I think in this image you can actually see one. That's where HCN likes to hang out. And I don't know how you would potentially align them across the cleft. But what we see in our imaging is that's where HCN likes to hang out. Those things are very closely opposed, close to this SR on the interior. And when you start making these little clusters and look at calcium transient, for instance, the calcium transient is most robust just interior to the cleft on either side. So what we're thinking is it likes to hang out, and this matrix may also serve a biomechanical purpose of kind of creating a very viscoelastic material around the cells so that there's not a lot of membrane tension and you get lots of caveoli, and that's where the HCN likes to hang out. So we've got lots of studies going on where we're pulling on cells, we're plating them on different materials and seeing them spread, and then actually tracking where particularly HCN4 and NCX1 move. Yeah.
I'm just wondering if you might have the opportunity here to create a model. So you mentioned that there were some gap junctions in there where you had the olfactic junctions. Could you create a model in a dish where you could kind of see the density of gap junctions and ask the question physically, what does it matter whether you do or don't have any of the gap junctions or a certain number of them? What I'm really thinking about is and Steve will probably want to answer this, I'm really thinking about the role of the EFAPs and the field on the gap junctions per se, and what, if anything, that's contributing to the electrical connectivity in the cells.
Yeah, so no, that's an excellent question. We've been thinking about this for a long time, trying to figure out the best way to do that kind of thing. Where we've kind of landed, and we've started building the reagents, and we haven't actually done any testing yet, is basically to make constructs that have wild-type chick connexins and mutated, basically dominant negative connexins, express them in situ, and then also put in at the same time an optogenetic tool, right? That'll let us paste the individual cells. So basically we'll have mosaic tissue that have some of these cells that basically can't form a gap junction with their neighbors, and optically paste that cell and see if the cells around it respond when the matrix is intact. So because I think the matrix is an important component. If we start to do that in the dish, I feel like it has to be a gap junction mediated thing. right? Because there's no sort of coating to surround them. So those are the kinds of experiments that we're trying to get to right now to be able to test that. It turns out when you overexpress a connexin in the tissue, right? You just go in and use this mosaic overexpression system. Overexpressing connexin 40 almost immediately kills the heart. So what we're having to do is kind of figure out ways to work around that. The dominant negative doesn't, but the overexpression one does. So we're kind of in a little bit of a catch-22 where we're trying to figure out the right way to do it. But I think it would be much simpler to coat something and then have the right.
You're up.
Just beautiful work, Mike. I'm just sort of gobsmacked. So, I guess the knuckle bit is parallel, like breaking it. It's, pretty clear the active side of the neuron. You're kind of thinking of the protocardial cells as active sites in terms of the relationship they pose on the myocytes. But I'm also thinking back to sort of some old-fashioned experiments that were done back in the day, sucrose gap experiments.
Yes.
And so on. Jose Gualife, I think,
Yes.
Followed up with some work in that area where he, inferred that gap junction coupling in the node must be important because, particularly sensitive to sucrose gap manipulation.
Yeah.
So I'm kind of wondering about, we sort of talk about some of these old-fashioned experiments and how to explain them, and maybe whether they could even be incorporated into your approach.
Yeah, so just to elaborate on for everybody else who may not be as familiar, there are lots of really elegant studies that go back, 30 or more years where they've looked at the influence of gap junction coupling, particularly on pacemaking cells. And there's a couple of different layers to that. Some of them are, they take a large strip of sinoatrial node, right? And within that, you're going to have, according to what we're seeing in development, some of the segments are going to be gap junction sensitive, right? Our inhibitors work on that. You really need to focus on the core cells. Beyond that, however, I also think to some extent that what we're saying here doesn't argue against gap junction mediated interactions. I think cells are pretty ambiguous about how they can interact to some extent. And what I think is showing here is this is a fairly robust system when gap junction systems go wrong, right? And this is not a criticism of the sucrose gaps studies, and there's actually a couple of different versions of those types of studies where they link pacemaker cells, sometimes even just through a completely independent system, where they've come to those conclusions, and they actually kind of worked out, how much conductance you need at like a nanosiemens level. But I think you have to be very careful about what area of the sinoatrial you're focusing on, and whether or not the reported slowdown in conduction, for example, that they're showing is actually meaningful, right? Because here, even if we slow down the periphery of the sinoatrial node, but there's still some conduction going through there, so the backup system may actually be important, right? So it's not a perfect answer, but we've been trying to think about, and each one of those studies has its pluses and minuses, how to actually interpret them given our data set.
Rachel?
So this is a little different training from like the others. Sorry. So I was wondering, when it comes to like developmental disorders and like heart defects are the most common type, are there defects in the SA node that like in humans that have issues with either this neuronal adhesion molecule or the perineuronal nets, or in patients who have defects in those disorders, do they have defects in their SA node as well as like neurological symptoms?
Yeah, so there's not a lot known about extracellular matrix defects in the sinoatrial node, particularly in young patients. So there's two problems there. One is if you have a real problem with your SA node, you don't develop very far, which is a challenge. What does happen is following corrective surgery for congenital heart defects, a lot of young patients end up with forms of sinoatrial node dysfunction very early, right? And the corrective surgery had no direct impact on the sinoatrial node. Now, we don't know why that is, but some of the things that we like to think about is we've changed the hemodynamics of the heart, and we may be activating these types of cells in ways that are atypical. They're changing their matrix production, making more collagen, for example, things of that nature, and they disrupt sinoatrial node activity. And so that's one way that we like to think about it. FLRT3 is actually very important during gastrulation in very early stages. So, even the mouse models don't make it very far, and there's not a flocks model that's been looked at, particularly in the heart. But even within that, within our data set, we see a lot of overlapping factors, right? So FLRT is one of them. We see a lot of synapse, single pass synapse transmembrane proteins being expressed by pacemaking cells. And we're not unique in that. People will often see these neural genes in the synovial node. And so we think that it may be sort of collections of these things that are interacting in ways that are similar to what we see at synapses and similar to what we see in like nodes of Ranvier. And they may be somewhat redundant with each other. So a single knockout is maybe challenging. I hope that answers.
All right, we're at the top of the hour. So just join me in thanking Mike once again. And